Okay, well, welcome. I congratulate you on being able to get here through this storm. We're from California, we're not used to this stuff, but it's great having this rain. I hope none of you have had any mudslides or any damage done. But anyhow, thanks for coming. As most all of you know, this is the medical series, and today is the first program for this semester. And the medical series, as you all know, is a great collaboration, and the Elks Club has provided this great venue for us for the past, I don't know, 10 or 12 years. We also use and have been offered and have used for all these years Morningside. And we have a medical program every week. On alternate weeks, we are here at the Elks Club at 9 o'clock in the morning. And the following week, we're at Morningside at 7 o'clock in the evening. And so we have a great series lined up for this semester. And Carol, we do have schedules. I hope you all have gotten a schedule of the medical series for this semester before you go home. It's also in the Blue Book. If you're an OLLI member, you have the Blue Book, and it's on page 56 of the Blue Book. So it's in there. Check it every week. There's some great subjects coming up. Today, we have a wonderful panel from University of California, Irvine, who are experts in the field of genetics. And as all of you know, and I'm sure you've all read, all the advances that are taking place in the field of genetics having to do with cancer, heart disease, congenital anomalies. All of these things now not only are being diagnosed, but are also being treated with genetics as the underlying foundation for the treatment. So we're going to hear about that today, and it is an honor to have this panel with us. Catherine Hall is a clinical professor of genetics at UCI. She's a genetic counselor there. She and her panel teach the freshman medical students in genetics at UCI. She's a graduate of UCLA undergraduate. She did her master's degree at the University of Washington in biology. She then did her Master of Science in Genetic Counseling at UCI, and a Master of Science in Genetic Counseling also at UCI. She is here with a panel of PhD candidates that have been writing and researching about genetics. So please welcome Catherine Hall and her panel of genetic experts. Catherine.
Thank you, Dr. Lawton, and thank you all for coming. This is one of our favorite things to do, is to talk to people about genetics. That's why we do what we do. In genetic counseling, we actually do genetic testing and things like that, but we also have an opportunity to talk to patients to help their understanding of genetics and genetic risks that they may have themselves or with other people in their families. So this is what we really thrive on. I was a high school teacher for many years and enjoyed talking to teenagers about all kinds of biology, but have really enjoyed most speaking about genetics with patients and in large groups like this. So thank you so much for having us here. I'm gonna say that we're gonna cover just such a small portion of what's going on in genetics today. Hopefully we'll answer some of those questions that you may have while we give the talk. So if you could hold your questions to the end, we'll be happy to talk about anything afterwards with questions that you're interested in about, but please note that there is so much going on in genetics that we don't know everything there is to know, so we'll do our best to answer your questions. I have 2 genetic counseling interns with me today. They're both training in genetic counseling. They will be finished in June and going off. There's a national board exam they take, and then they can practice in genetic counseling. They are required to do a thesis that requires research, so they're in the throes of doing that as well. It's a short program of only 2 years and a whole lot to do. So I'm really glad we're happy to come and give their time as well to come and speak with you guys today. Jessica is one of our panel members here, and she got I can get this right, a bachelor's in biological sciences from the University of Notre Dame. She is finishing up her PhD. I think she just mostly has writing left to do at the University of Washington in evolution and development of post-embryonic melanophore lineages.
Genetics.
So there. It is genetics. Has to do with genetics. And then she'll finish her master's in genetic counseling here in June. And then JC here got her Bachelor of Science in Molecular and Cellular Biology with an emphasis in bioethics. Yes, so it's a really, both outstanding students. We only allow 6 to 8 people a year into our program, and we have hundreds of people that apply, so we take the best of the best, and we're just very grateful to have them both. So I'm going to turn the program over to them and let them talk to you about all of the interesting things, and then we'll answer questions at the end, okay? So thank you so much again for having us here and coming out to talk with us.
Okay, can you all hear me? Okay, great. Technical difficulties. So as Kathy said, I'm JC. I'm going to start us off today with a quick outline of what we plan to discuss today. So we're going to start with talking about what are genes and how they're passed down and what happens when there's a mutation in one of our genes. And then we'll talk a little bit about cancer genetics and what we do in cancer genetic counseling and give some case examples. And then we'll also touch on direct-to-consumer testing and the pros and cons and what information you're receiving from those direct-to-consumer tests. And then we'll briefly touch on some new advances in genetics and genetic medicine and personalized medicine. And as Kathy said, we'll have time for questions at the end, so please just hold your questions until then. So to start off, we'll talk a little bit about genes and what they do in the body. So hopefully within the next 5 minutes, we'll be able to understand this diagram, but we're going to take it one step at a time. So to start off, our bodies are made up of trillions and trillions of cells. And in all of the cells in our bodies, we have our genes, which are packaged onto chromosomes. And this is what chromosomes look like in one of the cells under a microscope. And based on the pattern on the chromosomes, we can line them up into pairs. We have 46 chromosomes total, 22 identical pairs, and then 1 pair of sex chromosomes. So females have 2 X chromosomes and males have an X and a Y. And for each type of chromosome, we inherit 1 copy from mom's egg and 1 copy from dad's sperm. And then the genes are packaged along these chromosomes. So we have 2 copies of each individual gene, 1 inherited from mom, 1 inherited from dad.
So now if we zoomed in on a chromosome, we would see individual genes. And these specific genes are made of DNA. DNA is just a combination of 4 different chemicals in different orders. And we abbreviate these chemicals with the letters A, T, G, and C up here. And the order of these 4 letters is what makes each specific gene kind of do its job in the body. Each 3 letters codes for a specific amino acid. And chains of amino acids make proteins. And proteins are sort of the worker bees of the body. They make everything function. from things like how we metabolize food, how we turn food into energy, how your brain works, how your heart works, what color your hair is. So really anything you can think of. Proteins are doing that job, and the DNA code is what makes those proteins. So this is how our DNA serves as sort of a blueprint for the cell. But what happens when there's a mistake in that DNA code? So mistakes are called mutations. And in this top example, in this position in the middle there, there's an A normally at that position. But right below it, you can see if a C were accidentally placed there, it changes the specific amino acid, which might change the protein's function. And if that protein is normally part of an important function in your body, that could lead to disease because now we've changed that amino acid, which might change the protein function. However, we all have changes in our DNA, so not every mutation or change is harmful or disease-causing. We're not clones of each other. We all have variation in our DNA, so not everything's harmful. There are other types of mutations too called, so the example below is a deletion example. So in here, one single letter was deleted, that A that is shown popping out there, and then that shifts the whole frame of those triplets of letters. So instead of changing just one amino acid, all the amino acids downstream of that position are different. So those types of mutations are more likely to be really harmful to the protein and more likely to cause disease. So putting this all together in the full picture, we all have cells that contain chromosomes. Along the chromosomes, genes are packaged, and we get half from our mom and half from our dad. These genes are instructions to make proteins, which then carry out functions in the body. Sometimes a mistake is made in DNA that can result in a non-working protein and cause disease, but other changes are harmless. So now that you've aced Genetics 101, let's talk about how traits and diseases are passed down through families. Some conditions are autosomal recessive, which means a person needs to inherit two non-working copies in order to manifest disease, to have symptoms. So for an example, cystic fibrosis is a pretty serious lung condition that is recessive. For a child to have cystic fibrosis, he or she needs two non-working copies, which means his parents each have one non-working copy. So in this picture, we're showing the parents each have one non-working copy, but one working copy of the gene that causes cystic fibrosis. So they don't have cystic fibrosis, but they're carriers for it. And for each parent, there's a 50-50 chance that they pass down the working copy of the gene and a 50% chance to pass down the disease, the cystic fibrosis causing copy. And we don't get to choose which gene we pass down. It's a roll of the dice every time. So it's not anyone's fault. But so for this couple, for two carriers of cystic fibrosis, there's a 25% chance for an affected child, meaning to have two non-working copies and have cystic fibrosis, a 50% chance for each pregnancy to result in a child who's a carrier for the disease like themselves, so they don't have cystic fibrosis but could pass it down to their kids, and a 25% chance to have a child with no cystic fibrosis causing genes. Another form of inheritance that we see is autosomal dominant. And for these disorders, for these disorders, you only need one non-working copy of the gene to cause disease. So, for example, this couple, the dad is affected with the condition. He has one working copy and one non-working copy of the gene. So for each pregnancy, a 50-50 chance to pass down the copy that causes disease, and 50% chance to pass down a normal copy. And there's other types of inheritance we won't get into today, like sex-linked and mitochondrial. But the point is just, these are not the only two ways genes are inherited.
So, given what we know about disease-causing mutations and how they get passed down in families, hopefully you can appreciate how important family history is for us in genetics, and how it can help us understand somebody's health risks. So here are examples of how genetic counselors record family history for our patients. It's called a pedigree. Men are represented by squares, women are represented by circles, and individuals who are shaded have a particular condition, and then the numbers are their ages. So now that we have some basics down, we wanted to talk a little bit more about cancer genetics, which is really a growing area of our field in recent years. So if we take a look at cancer as a whole, we know that about 5 to 10% of all cancer is hereditary, meaning somebody's born with a mutation or a non-working copy of one of their cancer genes that increases their risk of developing cancer in their lifetime. Another 10 to 20% of cancer is what we call familial, so meaning we might not find an exact change in one of their genes that increases their risk for cancer, but we see cancer clustering in a family, so there might be smaller genetic effects and environmental effects that contribute to somebody's risk of getting cancer. And the remainder, and really the majority of all cancer, is sporadic, meaning it happens by chance due to environmental factors and age. So if we look back at those pedigrees I showed you before, you can see now how maybe the first example is a sporadic cancer in one individual. The second might be what we call familial cancer. So there are multiple individuals affected with cancer, but they're of slightly older age, and there might be a combination of genetic and environmental factors contributing to their risk. And the last might be an example of somebody with an inherited cancer syndrome in their family. There are multiple affected family members in multiple generations, and we see cancer at much younger ages in these individuals. So to understand how our genes affect our chances of getting cancer, we first need to understand how cancer comes about in the first place. So you might have heard all cancer is genetic, but not all cancer is inherited. So some of the jobs of the genes in our bodies is to help protect ourselves from turning into tumors or cancer. These are our cancer genes. So over time, we might be born with 2 normal working copies of these cancer genes, but over time, one copy might develop a mutation due to time and exposure to different environmental factors. But we still have one working copy that prevents the cell from turning into a tumor. But as more time goes on and we're exposed to more things, and these exposures like smoking, diet, age, sun exposure, and weight build up and can cause a second mutation in our working copy. So then we're left with no working copies of this cancer gene, and the cell can develop into a tumor. Now, if we look at individuals who have a hereditary cancer syndrome, they are born with a mutation in one of their cancer genes already. So again, as more time and environmental exposures occur, they might lose function or acquire a mistake in their second copy, meaning the cell is no longer working as it should and it can develop into a tumor. And you can see here that we have one less step than the sporadic cancer example. So this is why we might see cancer in younger ages and in multiple family members when there's a hereditary cancer syndrome in a family.
So now we'll go over a case example for what, when people come to see us for cancer genetic counseling, what will we do with that patient? So in this case, we have a 42-year-old woman who was referred for genetic counselor by her doctor because she was diagnosed with breast cancer at age 33, which is quite young. So she and her doctor want to know if she is at risk to have a hereditary predisposition to cancer. So the genetic counselor would start by asking questions about her family history because it's such an important tool for us. And here we have the patient with the arrow next to her she's 42 now, she was diagnosed at 33 with breast cancer, so she's shaded in because she's had cancer. Her father had prostate cancer at age 64. His, maternal aunt had ovarian cancer, diagnosed at age 65, and her mother had breast cancer at 44. So the genetic counselor would look at this family history, determine that it's high risk for breast and ovarian cancer syndromes, and then make genetic testing offers based on that. So the things that stick out for us as genetic counselors is how many people are affected with breast and especially ovarian cancer, because some of the genes that go into both of those, they share genes in common. Also, ovarian cancer is pretty rare. About 1.5% of women will have ovarian cancer in their lifetimes, which sounds like a lot. In the world of cancer genetics, it's a low number. And so the fact that we're seeing this somewhat rare cancer in the family makes us wonder, was there some sort of genetic element that helped push the chances of her getting this rare cancer a little bit higher? So then the genetic testing we might offer would be a sequencing panel of 17 genes. I listed them up there, don't memorize them, we won't go into all of them. So this is a sequencing panel for 17 genes that increase your risk for breast cancer. So this means the gene is reading the DNA, so the A's, T's, G's, and C's, and kind of looking at that spelling and order of each of those 17 genes and looking for mutations. And there are 3 possible results that can come from each gene that is sequenced. So the lab could come back with a positive result, meaning they found a mutation in one or multiple of these genes that increases risk for breast cancer. The result could be negative, meaning they looked at the spelling of the genes and didn't find any changes that are known to increase breast cancer risk. And then there's this third tricky result called a variant of uncertain significance. And that just means the lab found a change in the DNA, but they're not confident, they're not sure if this change causes cancer or if it's totally harmless, like that harmless variation we all have. And because the lab doesn't want to make a call either way without enough data, they'll just label it a variant of uncertain significance. So it's information we have, but can't provide medical advice on. And with time, we get more data. Sometimes these get reclassified to something more discrete. Most of the time, they get reclassified to harmless, but sometimes more research comes in and it turns out, oh, other people with the same change also had breast cancer or ovarian cancer. And then we would give advice based on that result. And something that's important to note is the more genes you test, the more likely you're going to get one of these sort of uncertain results. So for some people, these VUSs are sort of anxiety producing without, and we can't do action on them. So people have to decide for themselves whether they're willing to take on that potential extra anxiety. And also most of the genetic testing labs data in this country is on Caucasian individuals. So people who aren't Caucasian are more likely to come back with a variant of uncertain significance. Because again, we're all different, but Caucasian people are more similar to each other genetically than other people. And just without enough data on underrepresented populations, we're more likely to get some of these variants of uncertain significance. There's just something to be aware of. And this pie chart in the corner is showing the results that come from this specific genetic panel. And this panel, again, is ordered mostly for people with high risk, breast and ovarian family or personal histories. And what's really striking is the biggest piece of the pie is a negative result. So 70% there come back completely negative, which might be sort of surprising, right? Because this lady had this really striking family history, it had breast cancer really young herself. Wouldn't you think there's some genetic factor causing it? And the answer is there probably is. It's just we don't know all the genes yet associated with any type of cancer. We only know what we know now. And maybe in 5 to 10 years, if a panel were to come back negative, maybe we'd have more genes we could start to test. About 9% come back with a positive result and 20% with that variant of uncertain significance. So our patient's test report comes back showing that she has a mutation in a gene called BRCA2. Maybe some of you have heard of it. Angelina Jolie kind of gave this gene a little more publicity. And BRCA2 causes an increased risk for breast, ovarian, pancreatic, aggressive prostate cancer, so this might explain her dad's prostate cancer diagnosis, and melanoma. The sample test report here shows the gene name on the left. Next to it, it would say the specific mutation. This is a sample report, so it doesn't have an actual mutation. We don't want to use a patient's real DNA or results. No other mutations were found in the other 16 genes, so it's just this BRCA2. So what do we do with this information now that we have it? Well, the genetic counselor would then look through the most recent scientific literature, clinical management guidelines for BRCA2 mutations. And this chart's really busy, and the point is not to memorize it or know all these numbers. But some of the major points here are that the general population risk for breast cancer is 12%, but with the BRCA2 mutation, her risk increases to somewhere between 38 and 84%, depending on what her exact mutation is. There's also the increased risk for ovarian cancer, so instead of 1.5%, the risk is 17 to 27%. And that can have some pretty major implications because, so for breast cancer, we can do increased screening like mammograms, which are not invasive. For ovarian cancer, we don't have really great screening. So a lot of times ovarian cancer isn't caught until someone's pretty far along. So one of the recommendations is, and it's completely optional, but to remove the ovaries to prevent ovarian cancer. And so the genetic counselor would talk about the pros and cons of doing a surgery like that. You know, does this woman want to have her ovaries removed? It can be sort of a part of someone's identity for some people. It also means that individual goes through menopause right after that surgery. So there's a lot to talk about with any of these results. And I also wanted to point out that this gene, I know we've talked about breasts and ovaries a lot. It also increases the risk for male breast cancer, aggressive prostate cancer, and pancreatic cancer, as well as melanoma. So there's important information for men as well. And now going back to the family, what can we do with this information? So we know our patient has the mutation. She most likely got it from her dad because her dad's side, we're not showing you the mom's side of the family, but her dad's side of the family has all that cancer history. So her father should probably see genetic counseling and get genetic testing to confirm that he has that same mutation, because once it's confirmed that he has it, his other two daughters and his brother over to the left each have a 50% chance of having the same mutation. Something else I wanted to point out is his mother, who likely had this mutation, never had cancer. So just because you have a mutation in a cancer predisposing gene, that doesn't mean you'll get cancer. It just means your risk is increased and there's extra screening you should do. If this woman had kids, we didn't draw any in the pedigree, but each of her children would have a 50% chance to inherit this BRCA2 mutation and 50% chance to not inherit that one. And because cancer is an adult onset condition, we don't usually test children for these types of mutations. There's no management changes we would make. You know, we don't do mammograms on 12-year-olds. There's no real major increased cancer risk at that age, even with the mutation. So once those kids are older and mature, hopefully adults, they can decide for themselves if they want to pursue genetic testing.
Okay, and so as I think I mentioned before, cancer is just one of several areas of genetics in medicine. As genetic counselors, we work in many areas. One is prenatal. So in a prenatal genetic counseling session, we might talk about genetic screening and diagnostic testing options during pregnancy. We discuss ultrasound findings, as well as other pregnancy risks. There's also what we kind of just call general genetics. And we can kind of break this down into different areas. One might be neurogenetics, so giving a diagnosis and helping manage patients with Huntington's disease or muscular dystrophies. There's cardiogenetics, so an example is inherited arrhythmias. So I don't know if you've heard news stories about a somebody in their teens or 20s who's, passing out on the soccer field because they had a heart attack, they most likely have an inherited arrhythmia. There's also the area of metabolic conditions, so looking at our body's ability to break down the food we eat into energy. Sometimes that's not working properly. It can cause genetic conditions. One example is PKU. There are other genetic eye disorders, as well as the area of pharmacogenetics, so people who might not, or certain medications might not work as well for them based on their genetics. And so we've talked a lot about kind of the clinical side of things and clinical testing, but we all know that there are more readily available genetic tests out there called direct-to-consumer testing. And there are many different types. We put some examples up here, but you can test anything from your ancestry to different, what kind of wine you'll like based on your genes, and other things such as, health traits, health and wellness traits, and how easy or difficult it might be for you to lose weight based on your DNA. So we wanted to kind of go through an example of, what exactly are these direct-to-consumer tests actually looking at in your DNA? So one example is a company that has testing for BRCA1 and BRCA2, the gene Jess was talking about, associated with a higher risk for breast and ovarian cancer. It's offered through this direct-to-consumer test, but this direct-to-consumer test is not sequencing the genes. It's not reading through all of the A, T's, G's, and C's in our genes like the sequencing panel that Jess was talking about in our case example. This test is looking specifically at 3 mutations in the BRCA1 and BRCA2 genes, and these mutations happen to be most common in people of European Jewish ancestry. So about 2.5% of European Jewish people have one of these 3 mutations, but not all Jewish individuals who have mutations in BRCA1 or 2 have these 3 specific mutations. There have been thousands of mutations reported in the BRCA1 and 2 genes. I mean, you can see how big the genes are up there. So just because you test negative on this direct-to-consumer test doesn't mean there's not a possibility of having a different mutation in one of those genes. So it's just important to know what these direct-to-consumer tests are looking at so you can be the most informed. Another important aspect of these tests, and it says all of this in the fine print, but you can see there's a lot of fine print, and not everyone reads through all of these, but it does, say that it's not testing for every mutation possible in these genes, and it also importantly says that the results should be confirmed in a clinical setting, and you should talk to your doctor before taking any medical action on these results.
So you should also be cautious of any quote-unquote positive results. There are a surprisingly high number of false positive rates. And there's a lot of new papers in the literature because direct-to-consumers, come out so rapidly. So, for example, this paper in a clinically validated lab repeated direct-to-consumer testing on individuals who had direct-to-consumer testing. So they repeated the sequencing and reanalyzed it to see, are the people who came back with positive disease-causing mutations, are they really disease-causing positive mutations? And what they found is that 40% of the time, the so-called positive mutations were actually false positives. And there's a number of analytical and technical reasons why that is a PhD worth of information. But the point is, you could imagine for these 40% of individuals who get their direct-to-consumer back saying, they have a mutation and a cancer predisposing gene, which is what most of these false positives were in. That could cause a ton of worry. I know I'd be pretty worried if I came back with a positive. And 60% of the positives were confirmed to be true positives. So the point is, if you do direct-to-consumer, one of those health reports, and you get a positive, don't immediately panic. bring these results to your doctor, or just tell your doctor that you came back with a positive result on one of these tests, and your doctor can help you set up appointments with genetic specialists to reconfirm that positive mutation. And then the plus side of meeting with a genetics professional is they can go through, if this really is positive, which family members do we test now? What management changes do we do? Do we have to change your cancer screening? So the take-home message for direct-to-consumer testing is these tests aren't diagnostic. There's different methods. So like the case example we usually do sequencing of the full gene. Whereas like JC was mentioning, some of the genes on these direct-to-consumer tests are just looking at specific mutations and sometimes very specific populations. So I could have a BRCA1 or 2 mutation, and it is almost certainly not the Ashkenazi Jewish one because I don't have that ancestry. But that doesn't mean I won't get that mutation. So these tests just require clinical confirmation. Another kind of tricky thing about the results that we didn't go into big detail about is many of the results they give back are risk factors. So for example, maybe it tells you have a 10 times risk over the general population to develop early onset macular degeneration or Alzheimer's, which sounds really scary. But maybe it's 10 times a really small number. So you're going from 0.5% to 5%. So it's just important to, again, bring down the alarm, bring your results to a doctor who can maybe help you. Don't make any major lifestyle changes before getting results confirmed. Don't get your ovaries removed because you have a reported positive BRCA2 mutation on just direct-to-consumer. Another thing that's interesting is your DNA is as private as you let it be. So I don't know if you've heard the story about the Golden State Killer. So there's, the point is they caught a serial killer. He didn't, submit a DNA sample. Police made a profile and basically matched up his DNA to a distant relative who had done ancestry testing. And I'm glad he was caught, but just know that once your DNA is out there, companies can link you with family members that you may not even know are out there. But direct-to-consumer testing can be really fun. So it can tell you, there's traits on there like, do you like cilantro or will you hate it? Because there's one gene mutation that should tell you the answer. There's things like those funny, what kind of wine do I like things? And ancestry can be really interesting to find out about. So yeah, there's a fun aspect, but just know what you're getting into.
So very, very briefly, we're just going to touch on some of the new areas of genetics and medical treatment and personalized healthcare. So we gave one example earlier of cystic fibrosis when we were talking about the autosomal recessive inheritance. It's the progressive lung disease that can also affect pancreatic function. But within the past few decades, the lifespan of people with cystic fibrosis has changed dramatically. In 1955, children with cystic fibrosis were not predicted to live past grade school. Now, 50% of the population with cystic fibrosis is over age 18, and lifespan might be into the 40s and 50s. And that's in due part to new medical technology and advances in medical care. But we also now have treatment and drugs specific to somebody's genetic mutation in cystic fibrosis. So we don't have time to go into this picture in detail, but you can see that specific mutations cause particular problems in the cystic fibrosis protein, and we now have some targeted medications to target that protein mistake specifically and improve somebody's lung function and increase their lifespan dramatically.
And then there's another class of genetic treatment that's in clinical trials mostly right now, where treatments provide sort of a working copy of the gene, so to kind of correct the disease at the genetic level. And one way this is done is by inserting a viral vector, so just part of a virus without the part that makes you sick, and instead swap in a working part of the gene. And then insert that viral vector with the working part of the gene into the diseased tissue. And then your cells have the machinery to turn DNA into protein, because we do it every day, every minute. And now it'll also make that working copy of the protein from the viral vector and corrected gene that we have in there. There's also, you've probably heard stories about CRISPR technology. And CRISPR is a little bit different. The goal for CRISPR is so they inject in sort of molecular scissors, is the way I think of it, that cut your DNA, and they're targeted to cut out the part that has a mutation or isn't working. And then they'll also inject in a corrected version of that gene. So the idea is to swap out the non-working copy with the working copy. It's not as easy as I just made it sound. There's issues about how do we actually get these things into your cells, especially if you think of neurodiseases where we have, the brain, the blood-brain barrier protecting the brain, lots of neurons. How do you get all this machinery into all of those cells? And there's also a lot of fear about, so we've inserted scissors that are supposedly targeted to the area we want to cut out. What if it's not as well targeted as we thought it was? There's a lot we don't know about genetics. And so scientists worry about, potentially cutting healthy parts of the genome. So right now there's clinical trials for trying to treat retinitis pigmentosa and Duchenne muscular dystrophy with CRISPR. And I'm sure there's others in the works and more to come soon. And I think here are some really great books we found. And, jot them down if you're interested. Especially She Has Her Mother's Laugh is a really great, really comprehensive, but not too in the weeds, sort of, about just genetics and life, and, medical, evolution, all kinds of things like that. And then this website up in the corner, Genetics Home Reference, is a website. But the rest are books, really good about human genetics. And we'll leave this up while we take questions. Oh, and remind us to repeat the questions. I'll give you guys in the front that job if we forget. Sure.
So if I want to get, for example, for myself, genetic testing, where do I find it, and approximately what is the cost? I have no idea.
That's a really good question.
So the
Yes.
question was,
thank you.
if somebody was interested in genetic testing, how would they go about doing that, essentially?
So
And
what's the cost?
So I just want to give you, if you want to write this down, where UCI Genetics, and if you're interested, if you think there is testing you want, or even risk that you want to assess in your family. You have cancer in your family, you've had cancer, you're worried about your children and what their risks might be. You can always come and see us. I'm going to give you our number. It's 714, and I can repeat it again at the end before we go, but 714-456-5792. Again, that's 714-456-5792. You can find us on the web at UCI Medical Center in Genetics. Typically, depending on the type of insurance you have, there may be a cost for a visit, there may not. There may be cost in testing, there may not. But all of those things can be discussed in the clinical visit. Now, if you remember, there was a panel that we showed you with many, many genes, and typically if you're going to do something like that, if that's the most appropriate, thank you, testing for you, if you have something like Medicare, there are specific guidelines that number of affected individuals in your family, how far away are they related to you, first-degree relatives like mother, father, siblings, children. And if you meet criteria for testing, then typically Medicare will pay for that testing. If you have a PPO of some sort, then usually you have a share of cost, things like that. Depending on the gene, believe it or not, and the size of the panel, how many genes you're having testing, costs can be, I've seen, this is not just cancer, but costs can be anywhere from $250 to $20,000. And your genetic professional can help you sort that out. If you have an individual in your family that has a known mutation, in other words, they got back those test results that said positive test result and they have the mutation listed there, typically insurance will pay for your testing because you are at risk now, because we know in your family there is a higher risk. And those targeted tests, we're only looking, I kind of look at it, I think of it as the first individual that is tested that has cancer or has a cancer family history, but they're the first one tested. It's like giving someone a map and saying, can you find the blue house somewhere in the United States of America? You know, you've got a lot of searching to do. If we know what the mutation is, so someone has been tested and we have the mutation, we say, is there a blue house at 2463 13th Avenue in Seattle at this zip code? So we know exactly where we're looking, we know exactly what we're looking for. It's a much more conclusive test and less expensive test because we're just looking for something specifically. That bigger, more expensive test is because there's many different kinds of mutations, and different labs have to set different kinds of technology up and run it in their lab to find these different kinds of mutations. But like I said, your genetics professional can help you sort that out. Okay.
Many of us have gone through marketing agencies to test our DNA.
Okay.
23andMe
Yeah.
and Ancestry.com and so on and so forth. Are their results any good?
So the question was, if you've had direct-to-consumer testing, are the results any good?
I put it that way to
Yeah,
get
sure.
involved.
How valid are they? How useful are they? I think that's what you're trying to get at.
Because I would use that to come to you folks. Say, 23andMe said I have the genetic disposition for muscular dystrophy. I don't worry about that at 76, but this would be something that I could say I've been pre-diagnosed.
Do you want to take that one?
Okay.
Yeah.
Okay, so as far as pre-diagnosis, so these are not diagnostic tests. These are kind
Closer
of
to the mic.
screening tests, just looking for risk factors. So I can give you some examples of emails that I have received from individuals who have had these kinds of tests, and they become, for instance, one of the things that the reports will talk about is risk for Alzheimer's. And you may have heard of the ApoE genes, and at 1, 2, 3, or 4, some are worse than others. In other words, your risk for developing Alzheimer's at some point in your life would be higher if you have a certain 1, 2, 3, or 4. And so I've had individuals email me and say, I was diagnosed with Alzheimer's by this genetic testing. And so I say, please come in and let's talk about it. And the first thing you need to know is that this is not a diagnostic test. This does not mean you will have Alzheimer's. But we can talk about what it does mean, and that's the risk over a general population risk for developing the condition. And we do know, we didn't get a chance to talk about it, but there is an early onset form of Alzheimer's, and then there's kind of a later onset form, and we know that the early onset Alzheimer's, generally we're seeing people develop symptoms in their late 30s, in their 40s, and there is a very strong genetic component for that. In fact, there are 3 genes that we know of that cause this, and if an individual had a mutation in one of those genes, the risk that they would develop early onset Alzheimer's becomes about 99%. So it's very, very high. Generally, these direct-to-consumer tests do not test those. These are, the ApoE genes are risk factors. So when we talk about things like, we'll just say diabetes, we know that diabetes can run in families. We'll see many individuals in a family with diabetes, so we know there's some genetic component to diabetes, but we also know that with a healthy diet and exercise, that you can reduce your risk of developing the condition. So it's kind of similar to those ApoE genes. We know that Alzheimer's, the later onset, has a genetic component to it, but it's just one part of a whole picture. And those conditions we call multifactorial. There are many different components that lead to the actual development of disease. So yes, those are helpful tests to do, the direct consumer. You can hear about kind of what your risk over the general population is, and we didn't get the paper, but there are several studies out that show people who know they have an increased risk for some of these multifactorial conditions, so they know, they're informed that a healthier diet or more exercise can help reduce the risk, in general does not lead to lifestyle changes. So either you're kind of motivated to lead that kind of a healthy lifestyle, or maybe not so much. I don't think direct-to-consumer testing is necessarily a bad thing to do. I think it can be interesting. I think, as they both said, is that it's just good to know what you're getting when you do that kind of testing.
Yeah. Go ahead. Go ahead.
Well, it's two parts. First off, the example that you had up there, you had the lady with the breast cancer. She was heterozygous for the mutation. Mm-hmm. What's the difference between being heterozygous and homozygous?
Great question.
That is a really good question.
And then prior to that, the other part of it is one of the first slides that she showed with all the chromosomes. Mm-hmm. You had 3 for chromosome number 18. What disease is that?
Did we really? Oops.
Good observation.
Good observation. There's 3
Oh,
issues.
18's a little one. I missed it.
We talked a lot about, I'm going to start with that question. So the question was, why are there 3 chromosome 18s instead of 2? So I'm going to start with addressing that question first. So we talked a lot about genes and how there can be a mutation very specifically zoomed in on this chromosome, on the gene, there's a mutation that causes disease. There can also be what we call chromosome conditions or chromosome abnormalities. So people can be born with an extra or missing chromosome. This, when the egg and sperm come together, if one has an extra or missing chromosome, that can result in a child with an extra chromosome. In this case, this individual has an extra 18, which is called trisomy 18. It's a rare chromosome condition that can cause severe intellectual disability or other severe medical problems. These individuals typically don't live very long, and that can happen when you have an extra chromosome. Another chromosome condition is Down syndrome, so an extra chromosome 21, and this is not as severe as this trisomy 18. It's typically more mild to moderate intellectual disability, sometimes other health concerns, but that's a more typical chromosome abnormality that we see and most people have heard of.
And then, we'll do heterozygote versus homozygote. So I'm going to use this as my example. Heterozygote is an individual who has one mutation on a copy of their gene, but on their other copy of that gene, they don't have that same mutation. A homozygote has on both copies of that gene, they have the same mutation. So I'm showing this picture because this kid here might have 2 of the same mutations for, I think our example is cystic fibrosis. So he would be homozygous or a homozygote for that specific mutation, whereas these kids in the middle here are heterozygous for that mutation because they have one, but their other copy doesn't have that mutation. And in the cancer example, cancer's a little tricky because it's not necessarily straight recessive and dominant for whether you get cancer. Having one copy of a mutation increases your risk for cancer, but doesn't mean you'll get cancer. So it's still inherited in a dominant way, 50-50 chance to pass down that mutation, but that doesn't mean getting cancer is 50-50. It's getting a higher risk for cancer is 50-50. I'm glad you pointed that out because I wanted to explain it, but thought we didn't have
And two copies.
yeah.
In other words, you're homozygous for the BRCA2 mutation.
Yes.
Does that further increase your risk versus
Do you wanna talk about that
being heterozygous
Yeah.
for that mutation?
Good question.
So that's a really good question. And actually, that happens. We do have individuals who have either two different mutations in their two BRCA1 genes. It actually causes a different condition. I don't know if you want to talk about that at all.
That's okay. It is, yeah, it's called Fanconi anemia, and it leads to childhood cancers, so even earlier onset, and other health conditions, really severe anemia, so severe blood condition as well. So it does, compound the risk and introduce new problems.
Different
Can I
conditions.
just throw one more
Yeah.
thing in there?
Oh.
And in some cases, not necessarily for this gene, but there are some genes where if an individual inherits both non-working copies, it's lethal. Either the pregnancy would not survive, or they may survive only a few days. So it kind of depends on the gene, when it's turned on during development, what tissues it's on during development. I mean, we like to kind of pat ourselves on the back that we know so much about genetics, but actually we've come a long way, but gosh, there's so much more to know. So that's another way that the family histories can help us if we see a lot of miscarriages. That may give us some idea that maybe there's mutations, more than one mutation going around in the same family. We see that a lot in families where there's what we call consanguinity. So cousins marry and have children. There are a lot of ethnic populations where that is the preferred method of coupling, that people within the same family come together and have children. And you can imagine that if there's a mutation in the family and those people are related, then the chances that each of those individuals carries that same mutation is much higher. Therefore, when they have children, the chances that their children will get both of those mutations is much higher. So that's something we also talk about when we look at family histories. And there are some, you may have read in the news at some time, I think it's been 10 or 20 years now, I think. There are two to know about it. But anyway, that there are some Orthodox religions where there may be a rabbi that looks at the genetics of the two individuals that would like to be married, and without telling them their genetics will say, yes, you can marry, or no, you cannot. And that's based on risk factors for offspring. So.
You mentioned the CRISPR technology in patients with retinitis pigmentosa, and what I was wondering is what is the age that they would intervene in that kind of situation, and also what would be the technique to get the corrected gene, editing the corrected gene into all the cells of the retina?
Okay, so the question was basically, how does CRISPR work? How do you get it into the tissue that's affected? And what age would you kind of implement this? For retinitis pigmentosa. I don't know the specifics about retinitis pigmentosa. Traditionally, with things like this, we'd want to treat as early as possible. So once these sorts of technologies are approved, which they're not out for the general public yet, the companies that will sell CRISPR for specific disorders, I would imagine would set an age when it's okay to do that, based on, what comes out of their clinical trials. What am I thinking of? Like off-target effects or risks there are. Ideally, we'd want to start early. This is coming from someone who is not in a clinical trial or knows what they're privy to. But when it comes to correcting genes and treating genetic disorders, you usually want to start earlier because a lot of these disorders are progressive, so they get worse with time. And as you build up damaged tissue, a lot of the times we can't, even if you put the right gene in there, for some of these, you can't reverse the damage that's already been done. So catching things earlier is typically better, but it would depend on that specific, pharmaceutical company and their guidelines. For how to get the sort of molecular scissors that are part of CRISPR in the corrected gene into tissue, like the retina. I don't know how they do it for the retina, but I think part of the problem with CRISPR is, is getting the technology only into the cells that we want them to, not extra cells. And I think, I don't know if Kathy knows anything about this, but I think a lot of them are by injection.
Yeah.
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 So the other thing that I'll say about CRISPR is some of the research that's going on right now, we're using CRISPR technology, say in mice that we want to have a disease that we're studying, so we can study how the proteins are affected. And so a lot of times now we use CRISPR to make animal models with the conditions that we want to study. Not necessarily are we ready to cure all diseases yet, because many times we still need to figure out the function and how the proteins are working and all of that kind of thing. And I will say that, I'm trying to remember, it was for a gene called NUBPL. It's a neurological, at least a neurological condition, and we were trying to make what we call knockout mice. So we were trying to mutate that specific gene of NUBPL, and we used CRISPR. I am not in the wet lab doing the bench work, but I'm involved in talking about the research and working with them. We were trying to make these knockout mice so we could start looking at, are there medications that can target this and all of that. And we only managed to get one litter that had the knockout gene in the right place. And if you looked at the genetics of the other pups, we had things turned upside down, put in other places, all over the genome. And so although it was great, we were able to get one litter. You can imagine that if you're looking at the embryos of humans and you want to put the correct genes in there, the ratios are not in our favor right now. So there's...
On the mice, were you doing it in the embryonic stage? Or?
Yes, we were trying to create, litters.
Quite a luxury.
Certainly, with that, I don't work in the lab again, and so, many people have different feelings about animal models and that kind of thing, but you can see just from this that statistically we don't have it down just yet. So there's still a lot of work to be done.
Yeah. And
Sorry, go ahead.
I was just going to add too that a part of the reason they've been so successful with retinitis pigmentosa is because it's localized to the eye specifically, and they are injecting this corrected gene into the retina of the eye. It's very localized, and that's why they've had some success. And it's not for every cause of retinitis pigmentosa. If you don't know what that condition is, it leads to progressive blindness. So it's very specific for mutations, but the clinical trials have been promising so far.
If you inject it into one area of the retina, how does the opposite retina have
You'd have to do
change?
both eyes.
If you only inject it into one area, though, how do the other cells in the retina that are regenerating all the time, how do they get the correct gene?
Yes.
So as our cells are kind of growing and dividing and making new copies or making more cells, our DNA is copying itself and dividing. So once we have the correct copy in, that correct copy is, that correct gene is copying itself and dividing. So and like Jess said, it doesn't make up for what vision loss has already occurred. It's stopping further vision loss in that case.
Okay.
Since the gene is in its infancy, really, how do you preserve for the future? What tissue would you want to preserve so that your family could look at it? You know, we kept blood from my mother, but we're not sure if that's the right one, or if it decays, or what. So what should we be doing for the future to preserve?
That's a good question. So the question was, if you want to save DNA samples for, whatever testing develops in the future, what's the best sort of tissue to do that for, and how do you do it? So right now, we typically, for patients that have this same concern, maybe, we didn't find a diagnosis at this time, but their kids might want to do testing 20 years from now. The easiest way is to do a blood sample, because we have DNA in all of our cells. And for the most part, that DNA is the same in every cell. So rather than, doing a skin biopsy or a biopsy of any organs, we can just give some blood samples. And then, at least for us, we coordinate with a commercial lab because UCI doesn't store DNA. I think their price right now is a little under $200, but that might change in the future. And they would store those blood cells, and that means also the DNA, in a freezer for, I think they do up to 99 years.
So can I jump in
And
really quick?
yeah.
So I just want to say that the blood sample goes, but they actually extract the DNA from that sample and keep that, and they have a lot of paperwork, but you can state in this paperwork who's allowed to have access to this sample. And believe it or not, just a single tube of blood gives so much DNA to preserve that there are lots and lots of tests. There's a lot of testing that can happen in the future based on that just little tiny bit of DNA that's stored. They used to say it was stored indefinitely. Now they say 99 years. I don't know where it will go. At some point it would be, I don't know, imaginable that they just sequence it and store the data.
Well, what's the best way to store blood, period, if you're keeping it at your house from,
Well, it probably depends on what you're storing it for, but if it's for genetics, I would say I wouldn't store the blood. I would send it to a company and have the DNA extracted, because the DNA can break down over time if it's not stored properly, and it's usually stored in big freezers that are not just like an at-home freezer. They're, minus 80 or
Celsius.
something like that. So they're very, very, very cold. So the company is called Prevention Genetics. You can find it online, preventiongenetics.com. You don't have to go through a genetic counselor or a geneticist to use that. You can go right online and you can see banking DNA is what they call it. And you can call them. They have a specialist there. She's wonderful. There's kind of one person that's in charge of all that, and she can walk you through that information. And it's particularly useful in families where the person who so for instance, I'll use Huntington disease, which I see very often. We know that there's an individual maybe in their 40s, so they're kind of at the time we might start seeing symptoms of this, but they don't want to be tested because they don't want to think about that. They don't want to know what's coming or not, but they do know that they want their children to have the option of testing later if they would want to know. So those individuals may choose to have their DNA banked and even put it in a will. So these people have access to my DNA sample to do whatever kind of testing they might want to do. So that's something that people choose to do now, because the testing changes so very quickly, and as we get to know more, there will be more options. You want to start way in the back, over there?
The first time I heard this was on a TED talk, and the guy was from MIT, and you spoke about life extension. You know, the identification of these diseases and of course the replacement of those diseases in the DNA. I'm wondering if we're headed in that direction, one. And two, what kind of research is going on, you know, in the United States? Will only the rich be able to, you know, to buy the tests and to begin to look at their life extension of 150 years?
I'll start by talking about research. So,
The question.
oh, and I'll start by repeating the question. Thank you. So there's a TED Talk, and there's research out there about using genetics to extend our lifespans. Some people saying maybe at one point up to 150 years. Are we at all headed in that direction? And then sort of what are the ethics of this? Is this going to be a technology that starts out being only for the rich, who will have access? And especially in this country, what kind of research is being done on things like that? So research out there, especially in this country because things are regulated, because we don't want to do research, you know, for the most part on human embryos. Most research is done on DNA from cell lines. So like a pool of cells from an animal or from animal models like Kathy was talking about with mice, or there's fish or fruit flies, just to see if we can identify genes that lead to longer life and things like that. And what was I going to say about that? Yeah, because animals, even fruit flies, we share a lot of the same genes. So even animal model research can be really helpful in grappling with some of the research in areas that are more controversial. We're not going to jump right to experimenting and trying to make humans live longer. And I think we're a long way from getting to really extensive lifespans. We just don't know all the locations of the genome that lead to aging. So aging is a really whole body process. It's not just, you know, one gene or one area of the body that is what leads to, you know, dying. So I think we just still have a lot to go on that.
Yeah, I'm going
I think
to
you
step in
can.
here too. So
Yeah.
you may have heard some of the research that was done where they were taking individuals in their hundreds, 110, and they were taking those individuals' DNA and comparing. So what is it that they have in common for these individuals that are living longer lives? That is a big process, and you can imagine that you could have a very big debate about causation versus correlation, right? So we may see lots of things in common with people, but does that really mean that that's making them live longer? There is a lot left to be done on that. But also we do know that, I will say, there are companies now where individuals can pay, it's right around, I think, $10,000.
San Diego longevity is 20.
$20,000 to have their genomes read, and then they come together with, they kind of have talks like this where everybody's then gets their results and they discuss the various parts of their DNA and what it means and the risk factors. And these individuals are usually healthy. So they're not looking for necessarily a disease-causing gene. They're looking overall, again, what is similar in healthy people that may be different than in people who have conditions. The other thing to say about that is quite some time ago, before we understood cancer like we understand it now, and we still have lots to understand, when people were living shorter lifespans, cancer was not as prevalent. So the ability to help people survive, you know, other conditions that were, you know, leading to shorter lifespans earlier, as people started to live longer lives, we find out that cancer becomes a leading cause, right? So it's really exciting that we can help people, individuals, fight cancer, go into remission, and not have to worry. But do we know by the time you're 110, if we're all living then, what's the next thing going to be? We don't know yet. So now we understand some things about the hereditary aspects of cancer. We're still learning about those even, but then these environmental factors, and you guys probably remember various chemicals and how they affected pregnancies, and what that did to limbs and fetuses, and so it opens up a big unknown for us. But as people begin to live longer, we'll kind of find out what the next frontier is going to be that we need to start looking into. Very exciting. That's why we love genetics.
Okay.
Go ahead, Justin.
Go ahead.
If a person is diagnosed with a genetic-based disease such as Alzheimer's or cancer, is it routine for the physician to request genetic testing?
Do you want to? Okay.
It kind of depends. So some conditions, such as Alzheimer's, if this condition starts to show, if the individual starts to show symptoms in their 70s or 80s, kind of later in life, when we know that the chances are likely there's not one single gene that's causing this presentation, probably not. They probably will not be someone that is referred to genetics. Now, that being said, UCI has a wonderful research institute called UCI Mind, and they work on all kinds of actually dementias, frontotemporal dementias, Alzheimer's, the list is very long. And they will actually ask for genetic testing of many kinds just to also look for those risk factors like ApoE, that gene that I talked about. Because we do eventually hope to sort out just how much genetics plays a role out of that multifactorial presentation of Alzheimer's. In other words, what percentage is genetic, what percentage is exposure, what percentage, until someone would develop symptoms. When we see it very early in the 30s or 40s, it's really mostly genetic, that presentation. But when we see it later, we know that there are many things that play a role in that. And so part of research, if you were in a research study, you probably would have some genetic testing, but the average patient would probably not be referred at that point. So it kind of depends on the condition too.
When conception takes place, do we inherit from our parents 50% from each, or are there certain 45, 55, or are there certain things that are stronger or weaker that are not passed on? For instance, let's say a nose, chin. You've got your mom's nose.
Yeah.
I mean, is that something stronger or weaker that's passed on in conception?
Who wants to take this?
I can take it. I would say that in most cases, we wouldn't say that it's stronger or weaker. There are very few, so parts of the body, your nose is not one gene, and so it's going to be a mix, and they're on certain chromosomes together, so you have a lot of genes on your mom's chromosome and those same genes on your dad's chromosome, so it's reasonable that sometimes you get mom's whole list of those genes, and those particular changes that they have in that set of genes from mom may overpower dad, but it could be the other way as well. But also we know that there is crossing over that occurs even between moms and dad. That's why siblings don't always look similar, right? Because there can be crossing over of mom's 2 chromosomes there is infinite variation that can happen. So we do hear a lot of times when patients come in and they say, well, my mom has this condition and I look just like her, so I must have it too, like even if it's a predisposition for cancer. And what we can say is that those are not linked in that way. Just because you look like one parent doesn't mean that everything about that parent you're going to also have. Those are very different.
You're reading a lot today about stem cell therapy. And to me, that's all genetics, because stem cells are a cell where the genes turn on and off and they can become whatever. So what is genetics doing to be able to, on an individual organism that's already whole, rather than putting in stem cells, about being able to turn on and turn off the genes that you already have that created your arm or whatever, or nerve, to be able to get those then to literally regenerate without going through the whole process of harvesting stem cells and doing,
Transplants.
putting them back.
Anyone, you want me to take this one too? It's a very good question. So we're talking about turning on and turning off genes. We know that, gosh, at every time in your lifetime, there are genes that are turning on and turning off. And can stem cell research also play a part in that? So we didn't talk, there are many other kinds of ways that genes work. Not just recessive to non-working copies, or a dominant inheritance, a working copy and a non-working copy causing a certain way to look. There's also a thing we haven't talked about called methylation, and that's the body's process. I think of it as having a light switch that can be turned on and off at certain times of development. A gene can be turned on, the methylation is turned off, or there's a whole system of feedback, negative feedback, positive feedback within the system around those genes that can turn that off. And you may have heard of a condition called Prader-Willi syndrome. So, and if you haven't heard of the name particularly, you may have heard of how it manifests in these individuals who overeat and overeat and overeat so much that typically they die very young of obesity, diabetes, heart failure because of the stress on the body. And we know where chromosome 15, there's a specific region, and you have to have one maternal copy of that gene in the region and one paternal copy to function what we say typically. However, if that methylation, which comes from mom and dad, for instance, if the methylation doesn't work and it looks like two dad's copies, or it looks to the body like two mom's copies, it causes a condition like Prader-Willi. We're still working on how necessarily to turn those parts on and off. We're not very good at that yet. We now know that there is methylation and that plays a role, and that's a huge advancement. I will say stem cell research is difficult. Well, stem cell research or stem cell treatment can be difficult because you really have high immunosuppression in individuals. You have to really suppress their immune system to introduce these foreign cells. And so you may remember, boy that lived in a bubble and all of those things. Because they don't have a good immune system, the mortality rate is very high. So there's a big risk in doing stem cell transplants. So you don't want the body to reject that. So we're looking at some of these other therapies like the vectors, the virus that doesn't cause the sickness but is going to insert these genes, because the immunosuppression doesn't have to be as great. It's a shorter amount of time and it's not as aggressive. So this seems to be kind of a good area of research that may even function better than stem cell therapy in the future.
Okay, maybe last question.
There's a family history of Alzheimer's in my paternal side of the family. he died of Alzheimer's, and so most of his siblings died of Alzheimer's. And so I'm concerned about what is my, future, is it? So what can I do to find out if I have the gene or if I
Sure.
go on a study or what?
That's a great question.
So the question is, if I have a family history of lots of Alzheimer's, what should I do? How do I find out what my future might hold? So the first thing I would do is make sure your doctor knows your family history, because even if, your general practitioner should be able to recognize whether your family history is high risk for having some major genetic component and be able to refer you to a genetics professional. I think that's my whole answer.
Well, and I would say, however, if you are concerned, you can come in and see a genetics professional, usually a genetic counselor. You wouldn't necessarily need to see an MD for that. And we'd, again, do just this, a family history. We'd see what individuals have the condition, when did it onset, what were the symptoms at onset, those kinds of things. And then we could assess, this is probably not the kind we would do genetic testing for, and you're probably at a slightly higher risk than the general population because you have many family members, so there's probably some genetic component. Or, wow, this is kind of striking, and maybe we should go ahead and test those high-risk genes for you.
Does insurance cover it?
So that's another really good question. It depends, again, on your insurance. Things like Medi-Cal, Medicare, things like that, it's more difficult to get testing. And Medicare and PPOs where there's no authorization required to do testing or to go see who you want to see, we will submit and ask for testing, and what almost always happens is go ahead and do the testing and send us the bill, and we'll decide if we're going to pay for any of it. They call it based on medical necessity. Probably it would not change your medical management at this time unless you had a very striking family history of really early onset, and so chances are they would say,
Later.
it's not going to change your medical management, so we're not going to pay for it. But, I'm always amazed. Things change so fast, and we can argue it, but we have to have a good reason to argue it. So I would say if you'd like to know your risk, the best thing to do is see a genetic counselor and then just get an idea from there. Yeah.
How do you have the idea on why you do that, if you do have it? Just like-
Well, so that's a whole thing we
difficult
haven't
about
talked
Alzheimer's.
about is pre-symptomatic testing for individuals. We kind of touched on it with Huntington's disease, but it's the same thing with Alzheimer's. And it's a very extensive set of counseling sessions, because once you know, especially with these what we call high penetrance genes. In other words, if you have a mutation in that gene, the likelihood of you getting the condition is 90% or higher. Once you know that risk, you can't unknow it. And so we really are very careful with how we handle that. I've seen, in my experience, I saw a patient, very lovely, she was probably in her 30s, that had a very striking history of ALS. Lou Gehrig's disease. And she already had twins. They were twin daughters, about 2 or so. She didn't want to know because she didn't want to live her life worrying all the time. But she also knew, based on her striking family history, that in order to get into clinical trials, you have to have the gene tested and you have to know your mutation. You have to know what's in your family. So what she chose to do was to have the testing, and I have it sealed in a medical record. In the event she starts to become symptomatic, that testing will be done already for her. So she does not know the results of that, and she does not want to know unless she needs to get into a clinical trial, which at that point, she'll know that she has it, and she just has the mutation, so she can, in her family, from time of symptom onset to death was about 9 months. And so, yeah, it's a very strong mutation. And so she didn't want to wait to have that testing done based on her symptoms. She wanted to have that ready to go in the event she developed symptoms. So those kinds of things we take very seriously and handle things a little bit differently. And some people have that testing and leave in their will, my children have access to that testing, but I don't want to know. So, okay.
Oh, phone number one more time.
Oh, phone number one more time. For UCI Genetics, it's 714-456-5792. And we do take self-referrals, so if you decide you want to come and see us, you can. You don't necessarily have to have a doctor refer you, but we also definitely want to have any records that pertain to anything that you're concerned about ahead of time.
Okay, one last question.
Along that same line, if we've had the direct-to-consumer test, which does not test your whole genome,
Right.
but I have uploaded, I myself, okay, I have uploaded to Prometheus, you know, would you be willing to look at that and interpret it for me? Is that part of your service? Or is Prometheus like, oh, I want to roll my eyes?
Well, it's not that, but that is an amazing amount of information. So I would appreciate if someone wants to come and see me about that they pick out some things that are particularly concerning and bring those reports. Please don't send me raw data because quite frankly, I don't have time to look through thousands and thousands and thousands of mutations. But if you have particular things that you're concerned about or that worry you, please bring those, make an appointment. We're happy to talk to you about those. Yeah.
Okay, great.